I'm a basic scientist. I'm a neuroscientist. I've been interested in brain degeneration for many years, and I've recently become interested in the utility of cannabinoid-directed drugs in the treatment of traumatic brain injury. All right, so I'm going to do my talk a little bit. I guess I can walk around a little bit. I'm going to do my talk a little bit backwards in case I get run out of time. I'm going to start with the introduction of the people involved in the research. I'm not going to go through all of these, but just I'm going to say that we couldn't do this research without a big team of collaborators and also without the funding sources. I will mention Tanya Rex. I use a focal blast, air pressure blast model of traumatic brain injury. Tanya first designed the system for use in ocular blast injury, and I modified it for traumatic brain injury. This is Marcia Honig, my major collaborator in all of this TBI research. And this is Bob Moore, who made the compound that I'll be talking about today, the CB2 receptor inverse agonist. All right. There. So in keeping with this backward idea, I'm going to give you the summary of what my talk is going to be about, and then I'm actually going to go into the data. So we use the CB2 drug, and we target, oops, got two, I wanted to do, come on, back, there. I wanted to use my pointer. We target microglial activation to treat TBI. Microglia become activated in traumatic brain injury. They perpetuate and exacerbate the harm that's done. And so by targeting them, we hope to improve the outcomes from TBI. Now, microglia make a lot of CB2 receptors, as it turns out. So that makes them easy to target with CB2 drugs. And anyway, we use a drug called SMM189. I'll typically call it SMM. It's a funny category of drug. It's not an agonist. It's not an antagonist. It's an inverse agonist. So it does the opposite of an agonist. I'll show you exactly what that means in a future slide. We use a focal cranial blast model, so only a small part of the closed skull, the shaved head. The skull is closed, the skin is intact, the animal gets shaved, and we put a high-pressure air blast against the side of the animal's head to produce traumatic brain injury. That results in a lot of functional outcomes and neuropathological outcomes that resemble what you get in mild traumatic brain injury in people, including visual, motor, and emotional deficits. We treat them for 2 weeks, treat the animals for 2 weeks, beginning just after the traumatic brain injury event. And that turns out to have a lot of benefit because it rescues, greatly improves the outcome. The deficits are reduced, and the neuron loss and axon injury in the brain is reduced as well. We have preliminary evidence as well that semi-daily delivery of this same compound in a mouse model of Alzheimer's disease called the J20 mouse model slows memory loss and helps maintain connectivity between neurons in the brain. All right. So traumatic brain injury has been in the news a lot, concussion, for a variety of reasons. It's been a very prominent issue in the sports world. This is Alan Schwartz, a writer for the New York Times, who wrote a lengthy series of articles in the New York Times about the dangers of repeated head injury in football and what this can do in the long term to the players, even when the injury that's repeated is what's called subconcussive. So this is now Bennett Omalu, who was portrayed by Will Smith in the movie Concussion. Bennett Omalu was the first one to discover this syndrome in athletes. He first discovered it by doing an autopsy on the brain of Mike Webster, this football player here, who suffered from profound dementia later in life. And it turns out that you don't have to play for 15 years as a center and get repeated headbutts to the people that are charging at you. You don't have to wait till you're 50, 60 years old. It also happens in younger players. This is Chris Henry, who, because of his career in football, it would seem, suffered a progressive degenerative brain disease like Mike Webster that's now called chronic traumatic encephalopathy, CTE. His symptoms were different. He lost control of his emotions, and he died due to an accident brought on by that loss of emotional control. This is Ann McKee, who's extensively studied chronic traumatic encephalopathy as well. And this is what you get with repeated concussion: normal brain, and you can see a badly shrunken brain. Now, the problem doesn't only exist for athletes. The problem also is very prominent in the military, where soldiers get exposed to explosive blasts once, twice, many times. But even if it's only once, they wind up suffering the consequences of that. They have mood changes, memory loss, so on and so forth. So it's an issue of concern. And this is why we ourselves got into the research. So this is the compound we use. And I won't say too much about it, except that, again, it was developed at UT by Bob Moore, the Bob Moore Research Group. It has not a fantastic selectivity for CB2 by ligand binding, But in terms of a functional assay for CB2 receptors, it seems to be highly selective for CB2 receptors, and it acts as an inverse agonist in a cyclic AMP reporter assay. And from in vitro and in vivo studies, we know what it does is it converts microglia from this harmful state called the M1 state, in which they exacerbate the injury. It converts them to another state of microglial behavior that's more beneficial and helpful. And in doing so, we think that's what's mediating our benefit with the compound that we use. Alrighty, now this is what inverse agonism looks like. So this is an assay called the cyclic AMP reporter assay. A certain kind of cell line called the hex cells are transduced with this reporter construct that allows you to see how much cyclic AMP the hex cells are making. You also then transduce the cells with either CB2 or CB1 receptors, and then you expose these cultures, different cultures, to increasing concentrations of this broad spectrum CB1, CB2 receptor agonist, and you get what you expect. You should produce a reduction in cyclic AMP because that's what cannabinoids do. When they act at CB1 or CB2 receptors, they reduce cyclic AMP production. So this is normal. But what SMM does, instead of reducing it or producing no effect, what SMM does is it increases cyclic AMP. So it does the opposite of agonist, so we call it an inverse agonist. It doesn't do anything, however, of note at CB1 receptors. So it seems to be selective for CB2 receptors and therefore non-psychoactive. All right, so we've tested it in vitro to find out how it acts, and this is the reason I got into the TBI research, because it has a sort of anti-inflammatory profile. So what we did here now is we have primary human microglia cells in a dish. What we do is we expose them to a stimulant called lipopolysaccharide, LPS. And what that stimulant does is it converts them to the M1 state in which they express a bunch of chemicals called cytokines. So in all these cases, you see the cytokines going up with LPS production. This means these microglia now have been converted to the harmful inflammatory state. And so what our drug, SMM, does, in all cases, it reduces the expression of this harmful stuff and is combating the inflammatory state. Well, we can also look at other markers. When microglia go into the harmful M1 state, they increase expression of various proteins on their cell surface called CD11b, CD45, CD68. Those are 3 different cell surface markers. They all go up with lipopolysaccharide stimulation, and in all cases, when you're exposed to SMM, those go down. Now, we have additional studies I don't have time to show you, in which we looked at cell surface markers characteristic of M2 microglia in vivo and in vitro. Those go up. So M2 markers go up, M1 markers go down. We think that's good. So this is now the mouse model of TBI that I just want to walk you through so you know what it looks like. So we take the mouse, we anesthetize the mouse, and this mouse is knocked out. We shave the left side of its head, and we put a little white dot over the shaved area. That's our targeting spot for the blast. The mouse then goes into this sled with a sort of a foam cushion insert there where its head gets cushioned. There's a very tight fit. He goes into a very tight tube, and so he's very snug in there, but he's cushioned as well. This tube with this big 2-centimeter opening, this tube goes into this wider tube here, and this wider tube is this one here, and it has a 7.5 millimeter opening. That 7.5 millimeter opening, we center the mouse. We can see the white dot centered in front of there. And then we center this contraption in front of this, which is a paintball gun. It's been modified to put out a higher pressure blast. And we bring the mouse a couple millimeters away from the barrel of the paintball gun, and we fire it once. And the mouse winds up then showing a lot of different kinds of symptoms of traumatic brain injury. This is the blast area. It encompasses the entire forebrain of the mouse. You can see the forebrain behind there in silhouette if you know any mouse brain anatomy. And this is the family of blast levels that we use. This is the peak, and well, this is the peak for different blasts that we use and the duration of each of those peak blast pressures. Alrighty. So then, now we want to model mild traumatic brain injury. So this is just a single blast. Even mild traumatic brain injury in people has consequences. One of the characteristics of mild traumatic brain injury in people is like if a physician does an MRI of the brain, somehow visualizes it, the brain looks just fine. And the physician is inclined to say, like, you're fine, there's nothing wrong with your brain, go home. But what's underlying that, though, is that the forces of the blast, or whatever the impact was that caused the concussion, have wiggled and disrupted the brain and caused stretching, compressive injury throughout the brain. And a manifestation of that will be axons will be injured and will break. So this is what broken axons look like. Whoops, I keep doing that. So this is what broken axons look like. So these axons here in the brain of humans who've suffered a concussion, shortly after the concussion they died, but this is what broken axons look like even in a mild traumatic brain injury. These are labeled for different kinds of markers. Doesn't matter what they are, but what happens is the axon should be this long, smooth thing. Instead, you see this beads on a string kind of phenomenon with the axon. Those axons are damaged, and what's happened to them is axons normally look like this schematically. They have inside them things called microtubules that they use to transport proteins back and forth between the neuron cell body and the terminal, and from the terminal back to the cell body. When axons get stretched, those, oops, I am never going to stop doing that. So anyway, they break. They break into these different spots. Now, they're transporting stuff, so it's like breaking a hose. So the fluid or the proteins that are collecting at those spots now can't be transported anymore. What happens is they pile up, and so those parts of the axon swell, and then they can even break, and the axon's dead. Okay, so we see that in our animal model, which verifies that it is a model of mild traumatic brain injury. So we blast on the left side. This is the optic tract that I'm going to be showing you. So we're going to focus on this because this is a schematic of the optic tract. The left eye here, which is the side we blast from, this is the left optic nerve, which at the level of the optic chiasm mixes together with the optic nerve from the right eye, mixed together, and then what happens is, in mice at least, most of the left optic nerve continues as the right optic tract. So the important point is I'm showing you the right optic tract. So in the right optic tract here, we've stained the axons now in these brown, straight, but wiggly things. Those are normal axons. Notice there's no bumps in them. So this is the marker we used. If we look after 3 days after TBI, you see that we have some nice smooth axons as well, but we have these dark bumps. The axons are broken, they're torn. This is a different marker now for visualizing those axons as well. It doesn't visualize as many, but this is a mouse engineered, so some of the axons express this fluorescent protein. And again, in this case, 7 days after TBI, you can see broken axons. So we can look throughout the brain, and we see these broken axons. So I need to check my time. I've got 7 minutes left. All right. So important point of note is that here in the optic tract here are microglia. These things, these cells you can barely see are the microglia. They're fine looking, they're wispy, slender, but after TBI in the optic tract, when they're surrounded by all these damaged axons, the microglia go into destroy and remove mode. And so they shrivel up, they become darker, they extend these thick, chunky processes, they change. So they're in the M1 state now. I'm going to move it along here and not talk about these other slides here. So we then start to look at functional deficits after TBI and the benefit of our drugs. So the next series of slides are going to show you deficits and functional benefit from drug. The drug being a CB2 receptor inverse agonist, this thing called SMM. So visual acuity, you can actually measure visual acuity in a mouse. There are tests for doing that. And if we normalize to 100% as normal in the animals that got no blast injury and got vehicle instead of drug, we call that 100%. But you can see then for the left eye, those animals are down, their acuity is reduced by about 30%. That's a significant reduction. In the animals that we gave drug to after blast, there is a significant improvement. Their acuity is significantly better. There's another visual thing that we look at called contrast sensitivity. It's very important in human vision, and that's improved as well. And if we look to see what's the underlying basis of this deficit and the improvement with drug, we find that the retina is about 10% thinner after the blast injury, and that thinness is much alleviated by drug. And in the inner retina, there's especially a lot of thinning that goes on. That's corrected. The inner retina is the part of the retina that has the retinal ganglion cells. All right, so now we also look at motor function. To take you through some of this and just make the overall point, I'm just going to focus on this particular illustration so I can get through the rest of my stuff. So anyway, so this is a spinning rod task. You can put a mouse on a spinning rod, a rod that increases its spin rate second by second. And mice kind of like that. It's kind of a challenge for them. And normal mice, if you test them before blast, normalize that to 100%, and then test them the day after blast, 1 week, and 2 weeks after blast, they progressively get better at the spinning rod task. And the task is to stay on the rod and not fall off. So they get better at that. And the animals, however, that have received TBI, they don't do as well as the animals that received a sham blast. However, the animals with drug, they do a whole lot better than the blasted animals alone. So underlying this, we found that there is a rescue of some of the motor structures of the brain. And these are other functional tests of motor function. Here we put the animals in an arena. They run around fast or slow, depending on who they are. But the bottom line here is that After TBI, animals run around more slowly. Drug fixes that. After TBI, animals have trouble navigating turns, and the drugs, this is the difficulty there, and the drug seems to improve their ability to navigate turns. We can also measure depression in a mouse. So I won't bother with saying how we do that and just get to the bottom line here. We measure depression over a 5-minute period in our test. The test is called a tail suspension test. The fact that this red line here is higher than the blue line indicates the animals with BLAST are depressed. And you can see the green, the drug, normalizes the animals in terms of depression. We can also look at fear. Just want to see how I'm doing on time. OK, so I can slow it down a tiny bit. So we also can look at fear in mice. This is an important thing for the PTSD-like outcome from TBI. So what we do is we put a mouse in a chamber. The mouse is sitting in the chamber. He sniffs around. He explores the chamber. And he's relaxed about being in the chamber. Then he hears a tone. He hears the tone. OK, the tone's not very disturbing. It's not loud. It's kind of, innocuous. But then at the end of the tone, there's a very mild shock that's not hurtful, but is nonetheless a shock you wouldn't want to experience. So that happens 5 times now. And what happens is the mouse becomes afraid when he hears the tone. He shows a mouse fear behavior called freezing. So after 5 trials of training, the next day we bring the mouse back in and we test to see how well he remembers the fear, how fearful he is in response to either the context alone or the tone. So in response to the context alone, mice actually generalized the fear to the chamber where they got shocked. The animals that got blast injury, TBI, but no drug, just vehicle, they are more fearful of just being in the chamber over the first 30 seconds of the test, and this somewhat reduces over the second 30 seconds of the contextual fear assessment. And then we play the tone all by itself with no further shocks. And we look to see how the animal responds to that. And we do that 12 times. And so over the course of 12 repetitions of this, what's called the CS, the conditioned stimulus, the animal initially shows fear if it's a non-blasted animal receiving vehicle, and eventually stops showing any significant fear altogether. The animals that got blast show a lot of fear that does not extinguish. The animals that got drug show intermediate fear, but they do extinguish. So this drug has alleviated the fearfulness after TBI. All right. Now, this is the part of the brain that's responsible for regulating fear. It's called the basolateral amygdala. This is the left basolateral amygdala. The basolateral amygdala in people too, we assume, but certainly in mice, contains neurons that are fear increasing and fear reducing. So this is an engineered mouse where the anti-fear neurons are visualized with this green fluorescent compound. As it turns out, we serendipitously discovered that after TBI, the anti-fear neurons were reduced in abundance. It seems like blast kills the anti-fear neurons, so therefore the animals are more fearful. Our drug, however, rescued many of the anti-fear neurons, and so fear was reduced. Now, we've also looked at electrophysiology to see how well brain connectivity is maintained after our drug is given after TBI. So this complicated-looking thing here is showing you communication between the hippocampus and prefrontal cortex. This is showing you sort of a simulation of a memory transfer system between 2 brain regions important in memory. The noteworthy thing is that when there is a moment of firing activity in the hippocampus, there is, in a normal animal, a co-occurring moment of activity in the prefrontal cortex. And that's what that blue peak shows. After TBI, that's gone. The prefrontal cortex does not follow the firing activity in the hippocampus. After drug, however, that is restored. So we found that the drug also is beneficial in terms of this electrophysiological readout of brain activity. Finally, now switching gears, I'm going to tell you very briefly about the work we've been doing in Alzheimer's disease, an Alzheimer's disease model. Here we treat the animals for about 2 months every other day with this compound, a lower dose than we use for TBI. And then we have been looking at a number of parameters, memory parameters as well as electrophysiology parameters. It improves memory. Mice show less memory loss. And this also shows what connectivity is improved. So this complicated electrophysiology thing is again showing you activity linked between hippocampus and prefrontal cortex. And if it's high here, that means there's linkage of activity. When hippocampus fires, prefrontal cortex fires at different frequencies here. We're looking at different frequency bands for where the matching occurs. So in a normal J20 mouse, which is somewhat of an oxymoron, it's an Alzheimer's model, so it's not really a normal mouse. But in a typical J20 mouse that has disease but no drug benefit, there's very poor communication between these two brain areas. One of the problems in Alzheimer's is that synapses are lost and brain areas don't communicate anymore. And so with drug, that connectivity is apparently maintained because functionally the prefrontal cortex is following hippocampus. So that's it. I made it through everything, and thank you.